But I want to, of course, introduce our first keynote speaker, Trey Ideker, who has done a lot of seminal work both in systems biology and basic understanding of molecular and biological systems with many moving parts, and in the computational viewpoint of treating or encoding such data as graphs and networks and manipulating those structures with interesting algorithms and computational methods. And so we're excited to have him here to kick off the meeting and talk to us about his work and vision. Thanks.
And I like the way you couch the meeting in terms of the future. I think certainly our lab, I like to couch our lab that way. Sometimes the future comes at the expense of the present, unfortunately, when nothing works. But I think it's always important to think about at least 5 years out. And that really is, in that spirit, the words I'm going to say today are mostly towards the future and somewhat grounded in the present, as you'll see. Okay, so first question, why do I start my talks these days with a picture of an iPhone? And the reason is simply that if you look at where we're going in computational biology, and especially my subfield cancer genomics, it's really towards a type of Siri system, except a Siri that you don't ask questions or just ask questions about, where's a good restaurant in Pittsburgh tonight, or where's a good sushi restaurant tonight. She can, of course, answer those questions already to some ability, but questions of a more biomedical nature. So what if you could ask Siri questions like, Siri, my patient cells have the following genetic mutations in PTEM and DAM1, and of course these days it's an entire genome sequence worth of mutations with hundreds of different mutated genes, not to mention all of the non-coding genomic elements to deal with, not to mention if you have a gene expression profile or other omics data and clinical covariates, and you'd like to take all of that input data and return phenotypes. And so what phenotype should I expect with regards to diagnosis or disease subtype? What phenotype should I expect, of course, with regards to patient treatment, as in a sense the holy grail of what bioinformatics is trying to do in this space. So you might be thinking that is a complicated and a difficult question to answer, and there's a reason. It's a subclass of a much broader difficult set of questions related to the genotype-phenotype problem in genetics. It's the classic question in genetics: how do you connect genotype to phenotype? So why are those kinds of questions hard? And there's lots of reasons why people cite for the genotype-phenotype interpretation problem being hard. But these days, perhaps the most celebrated of those reasons is what's called genetic heterogeneity, or just heterogeneity, period. And with regards to genetics, what that essentially means, just to summarize up a large body of work and findings, is every patient's genome is a snowflake, unlike any other genome ever seen. That's why you're unique. And the same is true for disease. So in cancer, the disease I'm talking about today, you have hundreds of mutated genes. A few of those have been seen before in patients that came before yours, and those are called recurrently mutated genes. But other than a few recognizable patterns, largely you're looking at 80 other genes, some of which have been seen in maybe a few patients, but are largely mutated rarely in your population. And then you have another set of genes which have never been seen to be mutated before ever in a patient. And so it's the failure to find those patterns in data that makes it hard both for people and machines to subtype patients into groups, make diagnoses, and make treatments based on those patterns. How do you find those patterns, essentially, when most events are rare is the challenge. So that said, there have been over the years a number of strategies, both bioinformatically and clinically, for dealing with heterogeneity among a patient population. And the one I'll focus on today is, before I get to the solution, let me just give an example of how bad it is. So here is an example of a ovarian tumor cohort where they have sequenced exomes for about 351 patients. So I think most people know what an exome is, but just in case you don't, these are just sequencing the genes, or the 2% of the genome that encodes for for proteins and looking for mutations there. When you're talking about cancer, these are somatic genomes, meaning they've actually sequenced the patient twice. They've sequenced the patient at their tumor site, or from the tumor biopsy, and they've also sequenced the patient's normal or germline genome. You subtract the two to look at the somatic mutations, or the changes that have been acquired, presumably to drive, or some of which, not all of which, to drive that person's tumor. And so in this dot plot, what you're looking at is for these 351 patients in no particular order, just focusing on one chromosome, chromosome 17, not at nucleotide resolution, but gene by gene resolution here. You get a dot for the gene on the column if the patient on the row is mutated at that gene. That's what this is showing, and this is from a well-studied now cohort from the Cancer Genome Atlas Project, which is the NCI's big effort to sequence cancer genomes, started maybe 10 years ago. So you might be wondering, did Trey draw a line here at gene 170 or so on chromosome 17? No, it looks that way, but it turns out that gene is p53, which is the most recurrently mutated infamous cancer gene, probably for all of cancer, certainly for ovarian tumors, and that's what we're looking at here. So that's clearly a pattern. Unfortunately, it's present in most patients, so it can't be used to subdivide or cluster the patients either, but it's at least a pattern. Other than that, you may be wondering whether there's any patterns in the rest of these points, and the answer so far has been not apparently, either to humans or machines. Okay, so this starry night has been hard to analyze. So that just gives you a sort of feeling for what I mean by heterogeneity. And now back to the weapon that people have advanced, or at least one of the weapons people have advanced for dealing with heterogeneity. And this is an old idea. It's not a new idea. And it's the idea that maybe diseases that are complex, like cancer, are not diseases of individual nucleotides or even diseases of individual genes, but are diseases of some higher order entities in the cell or the tissue or the patient, which sort of aggregate those mutations into common features. So what was a rare feature at one level of physical scale in the cell now will become a common feature if you, either put on fuzzy lenses or simply think about it as a multi-scale problem and now deal with objects that are containers for many genes. And those are largely called pathways in our space. And so here is one example of where pathway knowledge has been applied to try to make sense of all of these rare mutational events in cancer genomes. This one, I'm going to switch tumor types on you. This is the TCGA's head and neck cancer analysis from just a couple of years ago. But I could have, in fact, used almost any of the 20 or so some odd TCGA papers that have come out in the past 5 years. Almost all of them have at least one pathway diagram somewhere attempting to do something like this. And so what is this? Here what they've done is group genes largely into 5 columns here. And so you can see one column is called the RTK-RAS PI3K pathway. One is cell death, one is immunity pathways, and so on and so forth. And within each column, they draw a wiring diagram of what genes and gene products they hypothesize are talking directly in some kind of communication or protein complex modular system to each other. And then inside of each of these boxes, you have the percent of tumors that are mutated in that component. So again, at the gene level, you have a few frequently mutated genes. Like here's EGFR at 15%. If it's an HPV negative head and neck tumor, they're already distinguishing between 2 known clinical subtypes, HPV positive and negative driven tumors. But then if you look, HRAS 5%, oh, okay, here's a 50%. That's not so bad. But the point here is that if you aggregate all of these events together and think about it as one super unit, then in fact you can catch 62% of patients. And that becomes, of course, a very significant recurrence in this type of cancer. So this idea is not a new idea that we should take a higher level or higher order view of these genomes. The question, though, is how do we really, given that idea, move forward? And there's 2 big challenges that we've been working on in our group, but I think there's others as well. Here are the 2 I'll focus on. So one, we can only assume, it's poorly characterized how much coverage we have of pathway space. And that's, in fact, not a well-defined question. But one can nonetheless think, what is our coverage of these connections that we'd want to know in order to analyze tumor genomes properly? And how do we experimentally begin to accrue all of the remaining knowledge we'd need? So one can enumerate different kinds of networks, and we'll talk about different kinds of networks in my talk. Actually, you have protein-protein interactions or direct biophysical interactions among proteins. You have transcriptional networks where proteins bind promoters and enhancers in DNA to drive gene transcription, and so on and so forth. Multiple kinds of networks, multiple conditions, multiple points in time, multiple patients. Nonetheless, one should think about, I would argue, about how we can achieve somewhat of a saturating coverage of some of those networks. Again, insofar as they're important for understanding tumor genomes and other disease genomes ultimately. So that's question one. It's very, very analogous to the genome sequence coverage problem, much less well-defined, but nonetheless analogous. How do we cover a human genome given we have these short length reads or reads of a certain length and a certain number of redundant reads over a given element in the genome? That was a very interesting problem in bioinformatics and still is to a certain extent. There is going to be and already is another set of interesting problems in bioinformatics about how do we cover pathway space. Okay. Question two is taking it now for granted that question one can be answered and we can accrue all of this information of pathway space. How do we then use that type of information programmatically for genome translation? And I'll try to touch on both of those challenges in my talk today. So to address these kinds of challenges, we recently applied and had funded last summer a NCI, or National Cancer Initiative, a center called the Cancer Cell Map Initiative, or CCMI. In fact, I'm just coming off the tail end of our annual meeting there in DC on the way back to San Diego for this meeting. Like I said, there are networks that one wishes to interrogate at a number of different layers. We don't claim that we're catching all of them, and even within a given network type, we of course have to be quite focused given a limited budget for how much we can do. But nonetheless, we have aspects of the center that are both mapping and attempting to use different kinds of maps in genome analysis. So one is transcriptional networks. I mentioned protein-protein interaction networks. There's also, I won't talk much about this today until maybe the very end, so-called synthetic lethal networks. These are logical connections between mutations, not physical connections between mutations. And then, of course, there's computational infrastructure the center has to support to do all of this, both hardware and software. Some of the faces of the professors and staff involved in the center are shown here. Nevin Krogan is my co-director. He's up at UC San Francisco, and he's an expert in protein-protein physical interaction mapping using mass spectrometry. But today, in interest of time, what I'm going to do is tell you just one story from the center's research related to transcriptional networks. And this is the recent work of Wei Zhang, who until very recently was a postdoc in the group, and Ana Bajorquez, who was a lab technician, but certainly performing at the level of a scientist, as I think you'll see in the study. So the context of this transcriptional network study was where cancer genome analysis really is going these days. And so I showed you some slides of papers from 3-4 years ago that were sequencing the cancer exomes, or just the gene encoding regions of those tumor cohorts. Now attention is shifting towards the full genome. So what about the other 98% of mutations that are impinging, raining down on the tumor genome? Are some of those drivers of cancer as well? Almost certainly the answer is yes. But the signal-to-noise problem is that much greater. It was already a difficult challenge to sort of figure out which mutations in genes are functional in driving tumors. It's much, much harder then to figure out which of those outside of genes in that sort of desert of knowledge are impactful functionally. So if you look in the TCGA again, in terms of what data are currently available, we have just shy of about 1,000 tumor whole genomes that are analyzable by the likes of us. And that covers about 12 different tumor types. So you can think about that as a quote-unquote pan-cancer data set across 12 different types of tumors. Now, the thought here is that to gain signal to noise, it would be important to run those data or the somatic mutations you find there through transcriptional network knowledge. Why a transcriptional network? Because here our premise is that some of those events in the non-coding genome are going to fall in so-called enhancers and promoters, which influence gene transcription. And so here, as the phenotype, rather than go all the way to patient survival or patient drug response or something like that, which you could do, we're going to use a much, much more intermediate phenotype, which is the gene expression of the genes in the tumors of those patients. This also exploits a very nice aspect of the TCGA, or I should say a very thoughtful aspect of the Cancer Genome Analysis Project, which is, in most cases, not in all, they didn't just generate one layer of data when they went after a patient's tumor. They generated multiple layers of omics data. And so in this case, we have not just the full non-coding genomes, but we also have the matched RNA expression profiles of those tumors. So that was the challenge. Could we begin to explain and connect how changes in the genomes or in non-coding elements over here at the genotype level give rise to changes in gene expression at the phenotype level through network knowledge here as the middleman, if you want to think about it like that. At the beginning of this study, which just came out about 4 months ago, we were cobbling this network knowledge together from a variety of different sources, like the ENCODE project, the Phantom project, the GTEx project, and so on. These are sort of well-known acronyms for those in the field. If you haven't heard these before, don't worry about it, because I'm going to explain everything anyway. And not only that, but by the middle of the study, we were able to sort of lay aside the sort of bespoke network we had constructed and instead go for what's called the Gene Enhancer network, because Doran Lancet's group at the Weizmann came out with this Vasiljevich paper, which does all that work for us in a more principled way, in fact, than we have been doing it for ourselves. So we just switched to Gene Enhancer. And from the abstract of their paper, what Gene Enhancer summarizes from a bunch of previous data and studies is about 285,000 enhancer elements genome-wide, and those cover about 12.4% of the human genome. So we're still not going to look at 86% or 87% of the human genome, but nonetheless, that's much better than just looking at genes. And the second thing that it provides in this resource is the connections, or at least putative connections, just candidate is the keyword here, between those identified elements and the proximal genes nearby in that locus that people think they regulate. Again, some of those and many of those are more speculative links than others, but nonetheless putative links. So that was the problem. And pictorially, we addressed it like this. So you start with a lot of non-coding sites genome-wide, about 35 million of them. If you look at those and just do a light filter on those sites that are recurrently mutated in just more than a few patients. This was not a p-value type of calculation, nothing significant here about this. We just said exclude mutations that don't arise in 2 or 3 patients. Okay, that gets you already down by 2 factors or orders of magnitude to about 2.5 times 10 to the 5th sites. Then, and those are the sort of taking the orange here down to the reds. So the oranges are all of the sites defined by Gene Enhancer, and the reds are those that are just mutated by a few patients or more in our data set. And then finally, if you then map those enhancers to genes via these green arrows, I don't know if anyone has a better laser. I think I'm just, you can sort of see that, but I can always use my finger, I guess, if I have to. If you then look at the green links into genes, then that takes you down by another factor of 10. That said, we still have about 2 times 10 to the 4 sites we can map to genes. And for those sites, we then did linear modeling against gene expression, or to try to explain gene expression, taking into account not just the presence, absence of mutations in each of these red loci in a patient, okay, but also a bunch of covariates that you'd like to account for. And I won't talk a lot about those unless you guys are curious, in which case I'm happy to. The machinery here is not particularly new for how we did that linear modeling. In fact, a project before us, I already briefly mentioned, the GTEx project, had applied almost the identical linear model as we did. It's linear regression. It's not rocket science. And one thing I will say is that if you're familiar with the term called expression quantitative trait locus analysis, or EQTL analysis, this is exactly the same as that, except in somatic mutation space. Normally, people try to connect SNPs in the germline genome to changes in gene expression in those same patients. Here, we're doing the same thing, except these are not SNPs. These are somatic mutations arising during tumorigenesis or later in tumor development. So that's how it works. You're trying to associate changes in the state of, or the mutation state of these enhancers with changes in the expression state of the linked genes. At the end of the day, by the time you run your significance tests and compute your number of significant loci at a given false discovery rate, we ended up with 193 loci that significantly associate with changes in RNA expression over this patient population. So is that a large number or is that a small number? Well, it's both. It's a small number because it's much, much smaller than the 35 million sites we started with. It's a large number because only one of those had been found before. So in fact, all of the analysis of non-coding mutations to a first order before this paper had focused on and found a single association that was very, very strong between mutations in the promoter of this TERT gene, which encodes a catalytic subunit of telomerase. The idea being that one way to drive forward cancer is to mutate the promoter that upregulates TERT and causes tumors, okay, or promotes tumorigenesis. So the other 192 of these were new findings, and presumably those new findings come about because of the vastly increased power you have statistically given the network knowledge. So given these 193 somatic EQTLs as a group, our first question was, are they interesting for real patient knowledge, meaning are a significant number or substantial number of patients actually affected at these loci? Because even if they're significant, if there's just a few patients affected, it's not that interesting a result clinically, certainly, even biologically. But it turned out, at least for the top 20 or so, that in fact these are quite recurrent in patients of different tumor types. So in this chart, oh, thank you very much. Let's see if this is better. It's much better. Thank you so much. So, so here are shown the different tumor types on the columns. On the rows are 20 some odd of these somatic EQTL findings. The way you read this is the colon demarcates the promoter or enhancer on the left of the colon that's impacted by somatic mutations in that example, and to the right of the colon, the downstream gene whose expression is affected by events in that region. And again, that can be a promoter in the case of TERT. This is relative to the transcription start site, or TSS, so that's just upstream of the transcription start site for TERT, going all the way 91 base pairs upstream. That's that region. But you'll see here for enhancers, you get these very large numbers, either a plus or minus of the TSS, and those are the enhancer regions. So the numbers in these red boxes, I think you might be able to make them out. The color choice here is unfortunate, but the numbers range from 5-6% all the way up to 70-something% for TERT, with many in the 40-50%, or at least 30-40%. And these numbers represent the fraction of tumors of a given type that have events in those regions. And so TERT, being the original finding published before us, is screaming. I mean, 76%, or was that? Yeah, 76% of melanomas are involving a non-coding insult or mutation in the promoter of TERT. But you see here we also have 43% for this element upstream of DHX34, depending on the kind of of tumor you're looking at. So that was the first question. The second question is, are these events predictive? Meaning, can we now go into a cohort we haven't seen before as a validation and find some of these same events? So for that, we then turn to a second large cohort, even larger cohort, in fact, of tumor whole genome sequences that was made available by a sister project to the Cancer Genome Atlas, and that is the ICGC, the International Cancer Genome Sequencing Consortium. The reason we didn't start with these is there's no expression for these particular patients, but we at least have the full genomes. So we couldn't find these events in this cohort initially, but we could validate at least the recurrence of mutations in those sites in the ICGC, and that's exactly what we did. This is a so-called QQ plot, which I won't go into the details. It's a common way of analyzing these data. I think many of you are familiar probably, but basically each of the events appears as a point in the space, and if you pull away from this diagonal, which is meaning this is the expected distribution of p-values in the new cohort for association, you can see that many, in fact the majority of these events are in fact found recurrently in the second cohort. The other kind of follow-up that one then wishes to do isn't validation or replication, but is functional validation, I should say. It's a different kind of validation. Functional validation, or finding or trying to explore whether these events have any function in cells, is a much slower activity, but the reviewers made us do 3. And so I'll show you one. So here is one of the findings, which was non-coding mutations upstream of this disheveled associated gene DAM1. There were just 6 patients in this cohort of 930 patients that had mutations there. But again, the power comes from these network connections that you know a priori. So here, 4 of those patients mutated, in fact, the exact same site, changing a G to an A at base pair minus 191 upstream of the DAM TSS. And suggestively, that change altered what looked to be a E2F family transcription factor binding site to an ETS family transcription factor binding site. Based on that, we then went into lab, and this is Ana Bojorquez who did this particular work, and made those constructs, both the wild type G and the mutant A at that position, upstream of a green fluorescent protein, so you can look easily at its effect on expression, and then use fluorescence cell sorting to look at whether that actually mattered for the amount of GFP you get, and in fact it matters a lot. That's what happens. So it's the x-axis here in the facts you should be looking at. Each one of these is a cell, and you can see there's a higher GFP expression by and large when you look at the mutant construct as opposed to the wild type. And then with Emma Farley and Stephanie Fraley's lab at UCSD, Stephanie Fraley in particular is an expert in matrix gels and cell invasion assays. She was able to show that if you then look at what happens when you express DAM1 highly in a variety of cell lines, this is one cell line, you get much more cell invasion / migration. And so this now provides a plausible pathway by which somatic mutations in the promoter of DAM serve to upregulate DAM's expression level, giving rise to an increase in activity of cell invasion pathways. Okay. So I am just over halfway done, and we are good for time. And now I'm going to switch gears, or I should say generalize, because I've talked about a specific kind of network, transcriptional networks. I've talked about a specific data set, these TCGA 930 non-coding genomes. But if you recall, the goal here was to come up with some kind of platform that would hopefully be a generalizable platform or a general platform for how you could analyze omics data of a patient and get out phenotypic predictions. Okay, and so the work I just showed you, I think, is representative of work in our group elsewhere, as well as the works of many others that try to inject some kind of prior knowledge about the architecture of pathways in cells as a way of genome interpretation. But we thought we needed more work along those lines. And the biggest thing that bothered us was the fact that when people draw networks, almost all of those networks are drawn as graphs, as graphical models of nodes and edges. And we know that by and large, pathways in cells in some cases look like graphs of nodes and edges, but in many cases do not. And in particular, we know that pathway structure in cell, or cell structure as a whole, as well as cell function, is deeply hierarchical. So we have this sort of well-known model in biology where changes in the genome, shown up here, I'm going to go top down, doesn't really matter which way you go in this hierarchy. Changes in the genome give rise first to changes in amino acids and sequence domains. Those changes in sequence domains give rise to changes in states of proteins. Those changes in states of proteins give rise to changes in macromolecular protein complexes, assemblies of multiple proteins, sometimes of many proteins. Those small and large complexes are players in signaling networks in a more fuzzy way in what people call metabolic processes. Those happen in organelles, cells, affecting the states of tissues, organs, and ultimately the patient. So how could we capture, or could we capture, this sort of very deep notion of the cell as a hierarchical multi-scale system? Why do I say multi-scale in the same breath as hierarchical? If you think just about the physical scale, what's the scale of a nucleotide? Yes, there's a Q&A part of this talk. What's the physical scale of a nucleotide? I'll take it. I like it. 10 to the minus 9 meters, let's say. What's the scale of a person? This is easier. 1 meter, right? So we have 9+ orders of magnitude that we have to bridge if you're talking about pathway knowledge. And so can we start to reflect this in our models was our big question. There's other ways we could have pushed on problems with networks as a model for prior knowledge about the cell, but this is the big one that bothered us and that I'm going to talk about today. And the reason we thought this one was particularly significant is due to the work of someone who should be no stranger to anyone in this audience, Herb Simon, a longtime CMU professor, won the Nobel Prize for Economics in 1978, but as you probably know, or as you should know if you don't, was also quite well known for his seminal contributions to AI at the time. And what's the bridging concept between those disparate disciplines, seemingly? And one of the main bridging concepts in Simon's work was this notion of hierarchy as the basis for all of complexity. And so he had a couple of sort of treatises that are quite famous. And the one that I just read a year ago, and I wish I'd read it 30 or 20 years ago, is a seminal work of his, The Architecture of Complexity. It's fairly short. It's still incredibly relevant, every bit of it, and I think has largely been not forgotten, but not read often enough in modern bioinformatics. And so just taking from, I'm just like 1 or 2 paragraphs into his introduction here, and I'm just going to quote, okay? So he's describing what this whole piece is going to be about, in his words, on the architecture of complexity. So the 4 sections that follow discuss 4 aspects of complexity. The first offers some comments on the frequency with which complexity takes the form of hierarchy, the complex system being composed of subsystems that in turn have their own subsystems and so on. We just saw that. The second section theorizes about the relation between the structure of a complex system and the time required for it to emerge through evolutionary processes. So yes, he's going to be talking about biology here, not as the entire focus of this piece, but one of his major examples is exactly the kind of cell and tissue biology that we're talking about here. And he argues in what's a oft-quoted sort of parable. He tells the parable of the watchmaker, how it would be essentially impossible, or certainly extremely improbable, for a watchmaker who did not use hierarchical modularity principles in their watch construction to ever complete the watch. Okay, and and then he goes on to talk about dynamics, hierarchies, and so on and so forth. But really, I'm going to focus on these first 2 points as our guiding principles here. So now we're talking. This is very much future, and the present is mostly Kool-Aid. I'll just tell everyone right now, but it does. You'll see something that sort of works now, but I think is absolutely the way forward. Okay, and is going to keep us busy for a long time, and maybe I can convince some of you to also get busy for a long time. We'll see. So our incarnation of given all those ideas about we have network knowledge, we'd like for it to be hierarchical, and we'd like to build these Q&A systems that essentially convert genotype to phenotype. Here's our best schematic of how we think that should look, and I'm going to then show you some fledgling efforts to build something of that sort. Okay, so first we think there's a module up here that should take data that we can measure, which is largely in terms of the architecture of pathways, focused in pairwise distances among objects, mostly proteins. So a protein-protein interaction network is just a type of pairwise distance or proximity measure between pairs of proteins in physical space in a cell. And many networks that we measure experimentally can be thought of as simply a co-location score or co-association in physical space in the structure of a cell. And so the idea is to somehow analyze all of those together in some integrative environment that would then not just end up with a integrated graph of nodes and edges, but would organize that information hierarchically. Okay, so that's a structural hierarchy set of questions. And then two, there's the question of can we functionalize that? And the goal there is once you can come up with those structures, then can we start to use those structures as priors in some kind of structure-guided machine learning? And I'll again give some work that's very preliminary on how we've thought about doing that. And everything I'm going to be talking about for the rest of my talk is really this paper here that came out also a few months ago and was spearheaded by Jinzhu Ma, a very, very talented postdoc in the group who used to be in Jinbo Zhu's group, who is also a speaker in today's program at some point. Okay, so first the structural question of how do we take network measurements, because that's sort of close to what the raw data give you, is sort of do two proteins touch or not, or more quantitatively, how close are these two proteins in some kind of data space. Right? That's basically what a lot of different omics measurements give you. Co-expression, how close those two proteins in co-expression space, another easily measured type of distance. So I'm going to summarize the first bit of this, just in interest of time. But the first thing we do is we take a bunch of different measurements of those sorts, a bunch of different network kinds, and we integrate them into a single pairwise protein-protein co-location score. And this is actually following the work of many other groups that have built integrative frameworks for doing that. Here we use a random forest where we simply put in each evidence type, protein-protein interaction, is there evidence for co-expression, is there evidence for synthetic lethality, yada, yada, yada. There's about 80 different features actually that go into these things. Ours is a homegrown random forest, but the ideas are coming out of groups like Edmar Kotz at UT Austin, Olga Troyanskaya at Princeton, and so on and so forth. So many people have worked on that problem. We're going to simply use those results and move forward from there. And so in these kinds of integrative endeavors, you then get one graph out to rule them all, and the quantitative score of a particular pair of proteins in that graph is simply summarizing many kinds of evidence. And so if you care about the kind of evidence, you have to then unravel it later. So for instance, ATG27 here and ATG10 have a high score, and you'd have to go back into the data and look at the features that explain that high score, and it turns out it's a very strong protein-protein interaction, as well as one of the most correlated gene expression pairs of genes in the whole data set, and that's why you get that red intensity there. Having done that, we can now look at the cluster structure in these matrices or networks, equivalently, that one can construct. So I think many people in this audience we're all very good at looking at these heat maps. At some point in your bioinformatics career, you become very good at looking at these heat maps, and already the eye can see that there is a cluster here, ATG 27 and 10. But look, ATG 10 is in an overlapping cluster with 12 and 7. 7 also participates in 2 clusters. So already that rules out 99% of bioinformatics algorithms to cluster data, which partition a set of objects. They don't allow an object to go into multiple clusters. And then look, already the eye can see evidence of multi-scale because there's a pink cluster, which is weaker but still quite significant, sitting above all of these smaller clusters at a higher scale. So the algorithm that you have to have, it is a type of clustering algorithm. And by the way, there's a whole physics community that also looks at clustering of graphs, and they call it network community detection. So if you look at the literature, community detection is basically written by a physicist, clustering is written by a computational scientist or biologist, but they're all looking at unsupervised organization of data, okay? And you can see that for the goal of constructing a hierarchy of components in cells, that algorithm needs to have a couple of principles that most algorithms do not. One is just the simple notion that a cluster can contain more than two objects. So if you simply build a phylogenetic tree, which is a binary tree, you will create all of these artifactual nodes in the hierarchy, which are having to deal with or just representing all these binary splits. But what you'd like to do, like in the case of ATG 7, 12, and 10, if these are all very similar to one another and equally similar to one another, is just merge them all at once. That's property one that you need. Property two you need is this idea of at any point in this hierarchy, not just at the leaves, but at any point internal in the hierarchy as well, one would like to be able to assign that object to multiple parent objects in the hierarchy, just as we see happening here with ATG 10. So the hierarchy we get after a particular algorithm we applied here in this paper that had those properties looks like this. So ATG 10 goes in multiple directions. And then sitting up above all of these smaller clusters, you see this mother cluster here, which is this corresponds to the big pink one here. Now, where do we get the names of these components? Or these clusters, okay, in the hierarchy? Well, so that comes by alignment to a reference hierarchy of knowledge. And luckily for us, there's a very popular reference hierarchy of biological knowledge. What is it? The gene ontology, exactly. So when you can, so by the way, this is not Go enrichment. This is direct one-to-one mapping of one ontology against another using ontology alignment algorithms. There also, we didn't invent anything. We simply borrowed from the ontologies field and took the best ontology mapper that we could find. And so the idea here is if you find a system whose gene content maps to a system or a term in Go with similar gene content, and also the parents and children of that system have good mappings to Go, then that's a good mapping. That's how that algorithm basically works. I'm not going to really get into it more than that. So having found those mappings, you can transfer the text over. And so ATG 113 came right out of Go. Core machinery of autophagy mostly, not exactly, but mostly corresponds to a term already in Go. The red highlighted systems were not in Go, but a quick read of the literature made it very evident that those had been missed by Go because they had multiple high-profile papers that supported, say, the ATG8 activating complex and the ATG12 conjugation system. So in that way, this is sort of a way to fill holes in Go. But much more interesting, you then have components like 183 that are neither in Go nor in the literature. So these are objects in data at some scale that have no known correspondence to anything we currently know as biologists. And those are where the new biological discoveries are going to lie. It's a little bit like an orphan gene. A gene just has a systematic ID. Those are genes where we don't know the function. These are just systematic IDs for protein complexes and larger subunits in cells for which we don't know the function. So the algorithm that we used is actually just a variant of clique detection. But it's what you might call online clique detection because we start, as you'll see in a second, I'll explain what I mean by that. So for this toy problem, and you can read about it here, and again, if I haven't made it clear, we don't claim we've solved any of these problems. This is just our early stab at some of these problems. I think there's a lot of room and need for better algorithms in this area, and especially efficient algorithms in this area. We are currently struggling to apply these kinds of ideas to a full human cancer hierarchy, by the way. Okay, we have a very ad hoc procedure that's gotten us there, but it's really unsatisfying. Okay, and there's a reason why this autophagy example here is small. All right, but the way it works is, and I should say the other reason why it's a nice problem to think about is one has the gene ontology. So what you can do is before you even look at a shred of protein-protein interaction data, just take the GO, flatten the GO to a graph. Okay, and so if you're not familiar, there's this thing called Resnick's semantic similarity. There's many semantic similarity measures people use to compute distances between pairs of genes through an ontology like Go. So you can simply flatten the thing and then try to reconstruct it with your algorithm, and that's kind of what we did here in this paper. So let's just say we have these 8 genes, A through H. Here's the real hierarchy. That's the gold standard you start with. You flatten it, just looking at distances through this hierarchy, and you get a adjacency matrix that looks like this, okay? And so the higher the number here, the closer in the hierarchy. So E and F are very close, so they have a very high number, 0.75. C and D are very close, so they also have a 0.75, and so on and so forth. Where A and H are very far, so A and H have essentially a 0 here in this scoring scheme, okay? So this is the simulated data, and we're going to ask whether and how, with that simulated data set, you can reconstruct that hierarchy. Notice this hierarchy does include the properties we want. It has systems or internal nodes with more than 2 children, and it has children with more than 1 parent. Okay? So how does it work? So we start with a very, very stringent threshold on this adjacency matrix. Okay, and in fact, we start with such a stringent threshold that you would not select any particular elements of that matrix. So that's sort of a threshold of 1. And then we progressively lower the threshold all the way to 0. And by the time, and at each threshold, you show only the edges whose scores are above that threshold in the adjacency matrix. And of course, by the time you're done, you have a complete graph. So that's what happens here at the top. Now, as you go along, you find cliques at each step. That's the basic idea. And so the reason why I call this online clique detection is because when one is finding the cliques, say in the graph here at the threshold of 0.5, one already knows the cliques that were found in the graph at the slightly more stringent threshold. And you can use that information to make the algorithm more efficient. We've started doing that, but I know we haven't completed doing that. Okay, that's the message here. So if you see, the first thing you do is you highlight edges C, D, E, F, G, H. Those form cliques and thus get recorded as modules in this hierarchy or internal nodes. And then you keep adding edges, finding more cliques, and finding more cliques. And it turns out if you follow this, you do in fact reconstruct exactly the input. Okay? So that gives you a feeling for how it works. And I need to keep moving in interest of time. We have some ad hoc procedures for dealing with error in the cliques, so we don't need perfect cliques, and so on and so forth. But again, please look at that paper. And of course, you can apply this with the right modifications to large data sets. And so this is what the entire hierarchy looks like for all protein-protein interaction data, co-expression data, and synthetic lethality data we can get our hands on for the budding yeast Saccharomyces cerevisiae. And this, by the way, will begin the final part of my talk where I only talk about yeast. I don't talk about cancer. And the reason is it's a smaller genome. We have much more data for constructing these hierarchies. And as you'll see in a slide or two, we also have much more data for training a neural network seeded by this hierarchy, okay, importantly. So that's what the whole thing looks like. And so now let's talk about, in the last few minutes, functionalization of this hierarchy. So now let's assume I can build this hierarchy. I will no longer question whether it's a good hierarchy or not. We can talk about that separately. And now the question is, given a structure, can I use that structure in a structure-guided learning problem to connect genotype to phenotype? Let me, as I transition to talk about that, though, let me just go back and just comment on the size of this hierarchy. So it is quite large. One can detect about 3,500 subsystems over about 12 layers. And this is just the budding yeast data set. Okay, so 5,000 or so genes. Those would be the leaves of this hierarchy. But then you can really get an exquisite amount of detail and depth in the data that are out there. For instance, without going into sort of too much detail here, I can reconstruct more or less the mitochondrion from protein-protein interaction data. Okay, that's like layer 10 or 11 at the top of this hierarchy, right? Cell splits into organelles, organelles split, and so on and so forth. So how do I do that? It's just a very, very big cluster. It's a clique that emerges near the end of the process, but it's a significant clique. Not the best way to find a mitochondrion, of course, not the original way it was found, but it's there. Okay, so now given that hierarchy, here's what we did next, or I should say Jinsu Ma did next. So we then turned to functional data. And the other reason we're using yeast here is because if you're trying to learn how to convert a genotype input into a phenotype output, yeast has by far the largest number of training samples that of any organism. There are at least 12 million of them. And how does that work? Well, at this point in time in budding yeast, scientists have deleted all single genes, all pairs of genes, and a growing number of triplets of genes. And in response to each of those genetic perturbation experiments, they've measured a growth rate. Not all of this work, but the majority, I would say just over half or some, maybe actually more than that, of this work is out of Toronto and published in the single publication Costanzo 2016. So how do you get 12 million measurements out of that sample or out of that study? It's because if you just think about 5,000 yeast genes and pairs of perturbations, there's 5,000 choose 2 of those, and that's 12.5 million. So that's how you get there. Okay, so it's easy to get to that large number when you systematically plow through these small k-wise gene deletion experiments. Of course, once you hit K of 4 or 5, there's no possible way anyone's ever going to make 4 or 5 Ys gene deletions. But at least for pairs and maybe triplets, we'll get there. So that's the nature of the training data. And we'd like to teach a machine learning system, a supervised system now, to take each of those single pairs and triplets of genes and as input and output a growth rate. That's the training. But now, importantly, we're not going to do this through a black box system. We're going to try to do this through a system whose structure is sort of pinned down to the structure of cell biology that we've learned through the separate structural learning step. Okay? And so for each, how does that look? So we build a deep neural network. And for each subsystem in that hierarchy, so each of about 3,500 of these, we assign a bank of neurons, not one, but typically a bank of neurons, to represent the state of that subsystem in this artificial neural network. And the other thing that we do is those bank of neurons have their inputs constrained, so their non-zero inputs must come from children of that subsystem in the structural hierarchy. So DNA repair, as an example, would have a bank of neurons representing the state of DNA repair. And its children would also have neurons representing their states, and only those neurons could be inputs to DNA repair, and its neurons can then be inputs only to its parents, and so on and so forth. And finally, the end of this thing is a single neuron representing the growth rate, okay? Or at least a bank of neurons whose first PC is the growth rate, or something like that. Okay, so I think I've given you enough information for how to sort of think about this neural network. So first of all, how well does it perform? So I wouldn't be giving this talk if it didn't have something interesting to say in terms of performance. And so here's the scatter plot or bars summarizing the ability to recover the measured growth from the predicted growth. This is in 5-fold cross-validation. Quantitatively is one number. How good is that performance? So if you simply take the Pearson correlation between measured and predicted growth of the model, it's 0.5. So is 0.5 a good number or a bad number? Well, because it sounds like maybe 1 should be your best correlation. But the thing we realized is that if one goes into one's own lab, and we have a yeast wet lab where we can go into our lab, and tries to reproduce a sizable number, even a moderate number of these single, double, triple gene deletion growth experiments, your ability to do that and recapture the original Costanzo publication is about 0.68. So it's possible a model could eventually exceed the accuracy of experiments It's not impossible, but probably too high of a bar for the beginning stages here. So we think the upper bound of how good you could hope or how well you could hope to do is around 0.65, 0.68. Nonetheless, that's the gap that we think one could try to cover. Now, so let me just quickly go through these bars. So these bars are previous attempts to address a problem that was close, not exactly this problem, but close enough that we could try to joust against these earlier approaches. And the answer is deep learning just blows them all away. So just the fact we switched to a deep learning framework, we think, bought us a lot. Okay, and why do I think it's mostly deep learning and not the structure of the system? Okay, because let me call your attention to this final bar. This final bar is what happens when we take the same deep learning framework and simply train a fully connected version that is not constrained by cell biological hierarchical structure. Its performance is identical within like 1 decimal, or 0.01, okay, to the actual or to the correlation we got. So the structure, unfortunately or fortunately, it's just the result. Didn't bias performance. It didn't lose its performance either. Okay. But just keep in mind, you can throw a deep learning machinery at this problem and do quite well. Okay? So what does structure buy you? It doesn't lose you anything, but what it buys you is interpretability. And that's the last couple of slides here, okay, before I end. So now, unlike the deep, fully connected neural network, I now can go back to each neuron and ask what neuron does that map to in terms of a cell structure. Okay? And so how is that useful? It's useful because when I have a genotype-phenotype relationship or prediction, observation or prediction, which is interesting or unexpected, I can then try to map that back to underlying molecular mechanisms. So here's an example of a double deletion, the genes PMT1 and IRE1 in yeast. Blue here represents a state that's lower than wild type, okay, that's driven down. Red would be up, but we don't see much of that here. And that gave rise to a cell state, growth, that also was quite deleterious. So it's a very slow growing cell that you get when you delete these two genes. And now to interpret why, it's fairly trivial. We simply now look at what are the neurons whose states are the most perturbed in that experiment, and they map quite cleanly to these two processes, ER unfolded protein response and its parent response to ER stress. So that's your molecular explanation that would explain that phenotype. So imagine now if this were a disease model, you'd be essentially explaining what are the underlying cancer pathways that give rise to a cancer phenotype. That's kind of where we'd like to head with this. But already in budding yeast, it gets interesting. So then that's a prediction. That's a sort of another kind of prediction of the model, that when you perturb these genes, you're affecting the state of this pathway, the ER-UPR pathway. So to look at that, it turns out we already had a collection of genetic interaction or genotype-phenotype data where instead of cell growth being the phenotype, people had looked at the state of ER-UPR as the intermediate phenotype. And it turns out molecular biologists have a promoter, the HAC1 promoter, that's widely accepted to be a proxy for the activity of that pathway. So what we then did is we took the first principal component of the neurons assigned to each subsystem in the model and looked at how well those principal components correlated with the actual measured activity of this pathway by the HAC1 promoter. And the best correlated subsystem was, in fact, ER unfolded protein response. And I think number 2 was its parent. Okay, and so this shows you the distribution of correlations. So the other subsystems, by and large, are not correlated in their states with this HAC1 promoter output, but this subsystem is. So that provides you reasonable evidence that the model got it right in this particular case. How to validate this systematically for any prediction of this sort the model made, I don't know. The other interesting type of question one can ask these kinds of models that are now coupled to cell biology is not just for a particular genotype-phenotype relationship, that's what we just explained in that last slide, but overall genotype-phenotype data you've used to train the model. What were the most informative or important subsystems for that phenotype? So that's like asking, what are the most important processes for growth? Say it was human height. What are the most important processes for human height? That'd be really cool if you could answer that question. Okay. So here, how do we do it? So now we go through I'm out of time, but you can ask me about it later. We have a way, and again, it's coming out of sort of the forefront of ways people are trying to interpret these deep learning models, of trying to assess how important a subsystem or this bank of neurons is for the model if you sort of remove them. That would be the sort of idea. And that's what you're looking at here, ranking those subsystems. And so it's a power law distribution of importances. And so which means you can make statements like this: about 20% of those 3,500 subsystems account for about 80% of the growth phenotype. And of course, you can look at which ones those are. Please check this out. Again, this is early days, but you'll see our early attempts to visualize and let you explore just we've exposed just the sort of simplest aspects of this model online in this website, dcell.ucsd.edu. OK, so I'm out of time. Let me summarize quite succinctly here. So I've argued that really to interpret genomes, we need, and just due to the heterogeneity, we're going to need prior knowledge to knit these granular events together into common patterns. And that knowledge largely these days is captured in terms of data in these protein-protein interaction networks and other kinds of networks summarized from different kinds of genomics. I gave you one example of that, which is applied to cancer, and it's a global transcriptional network we use to link mutations to gene expression. But then in the last half of my talk, I tried to generalize that concept hierarchically and use those hierarchies to seed neural network models for prediction of genotype to phenotype. The last thing I would say is we definitely need to recruit smart people to work on these problems. So I'll give a shameless plug for open positions in the lab. So with that, I'll take any questions. Thank you.
Terrific. Thank you. Questions?
I have a question about how well these methods work with dynamic data. So obviously a lot of what's out there in large scale is sort of single time point association.
Yeah, before after perturbation. Yeah.
And yet, I think we now know that that obviously is a very shifting landscape, and for some diseases, obviously cancer is important, but let's say I study inflammatory diseases, especially acute inflammatory diseases, where things are changing very fast. And so what you're looking at now is potentially meaningless in 8 hours and 24 hours, and yet people want to be able to make predictions about outcomes using this kind of data. Can you comment about how these methods scale or work with?
Yeah, sure. I mean, in this case, I wouldn't say the predictions are meaningless, and I mean these are like steady state predictions essentially. So, but I would totally agree with your point that we should think about dynamics. Other than acknowledge that we're going to have to think about dynamics, I have nothing intelligent to say. Maybe just this, that I think for one route forward, look at neural networks and trends in just neural networks as a whole. And so recurrent neural networks would come to mind as a framework for dealing with dynamics, right? It's all most problems neural networks address aren't dynamic right now, and the few that I've seen are recurrent neural networks. So maybe that gives some indication of how to move forward when you have a sequence of events in time.
Very beautiful talk. Thank you.
Thank you.
I'm asking the obvious, but it's still cool to hear. When you have your fully connected deep learner, how similar is the structure the deep learner brings to the prior?
Yes. So the answer is the structure. If you look at the fully connected one, the structure changes completely. If you look at different bootstrapped versions of your, if you just it's not stable with regards to slight perturbations in the input-output training. Across validations, they're totally different, for instance. And so here's what I take home. Tell me what you think. Here's my best guess as to what's happening. The optimization space is extremely flat. There's potentially many minima, a local minima. They're all equally good. Or there's a lot of ways to have a correlation of 0.5. Many different structures. It finds one. Biology has found one. We want the one biology has evolved.
Okay, we can debate that.
Right? that's how I'm thinking about this.
I think from a mathematical point of view, you have just a highly overparameterized.
We can agree.
Problem.
We can agree.
And I don't think biology places overparameterized systems, right?
Well,
I think also the question is how, and you very nicely acknowledge that, right? I mean, the question is how good is your measurement now to actually lock in, right?
Well, so maybe so what we've done is crash the pendulum all the way to the other side and say we're going to nail down all of these parameters. Even then, of course, it has a lot of parameters to adjust in terms of it can adjust all of the weights from a daughter to a parent in this hierarchy. That's a lot. Okay, even then, but that's almost certainly over constrained. So can we come up with environments that do joint structure function learning at the same time? That's got to be the way forward.
So this is kind of piggyback on that. I think you may have answered this a little bit. So if you were to do a comparison where you didn't constrain the structure and it was fully connected, you were saying that when you were inspecting those neurons and maybe what they were enriched with, because you can also do that, like you said, with inspecting some methods, like you develop one, but there's other methods right now to inspect what the neurons are.
Right, but you don't know what those correspond to in terms of a biological
So
structure.
you couldn't find any relation at all, or they varied a lot between?
Well, no, and they're different from fully connected network instance to instance to instance. They're more or less different, and so, yeah, not sure where you'd go with that
So,
for
like,
interpretation.
we've done some experiments ourselves with something similar, but using like gene expression data, not sequence data, and
Yeah.
we were seeing something similar to what you found, where the fully connected was performing sometimes probably better than the one that we had constrained with some sparser architecture.
With some regularization, yeah.
So we tried, okay, let's do some really strong regularization on the dense architecture. We were finding sometimes that we would see some of the nodes learning some pathways or some terms that were really relevant or activated for the particular phenotype that we were looking at. Sometimes we see this.
Well, and we should talk offline about how big that network was. I mean, here, so if the network's small enough, I think you can make some traction with that. But here we had 3,500 subsystems, each which had 5 to 10 neurons over 12 layers, and the fully connected network was as complex. And so it's hard to know where to go with that without some grounding in cell biology. Okay, thank you.
Yeah, let's thank Trey again.